My name is Jade Jaffar, and I'm a senior scientist working out of the Department of Respiratory Medicine. What I do is I kind of wear two hats. We both bank biospecimens from patients undergoing transplantation for lung fibrosis, and we also develop commercially important procedures in order to take novel therapies into the clinic. Lung fibrosis is a chronic respiratory disease that attacks the gas exchange units in the lung. And what happens is, over time, this damage forms scar tissue, which then prevents the ability of the person to breathe. There's no current cure for this disease. However, there are a couple of therapies that are able to slow down the disease progression. However, we're still looking for more effective therapeutics for our patients. When we perform clinical research, especially translational research, we really need access to human specimens. The best medicines are going to be developed using human specimens, and this is inherently difficult to obtain for many researchers. So what we try to do is accelerate the process by which we can access this tissue and share it amongst a lot of academics. So when the patient undergoes a transplant, instead of donating their tissue to one group, they would donate it to our bank, and our bank would be able to distribute that tissue to multiple research projects. So my research involves collecting this tissue and processing it in such a way that it could be used for downstream experimental applications. But we also derive single cell cultures from this tissue, and that allows us to test drugs in a dish that would allow us to see whether or not any new therapeutics would be potentially useful for our patients. There are currently in development for lung fibrosis something like 50 different compounds. And in at least the last 20 years, we haven't gotten very many that are useful. So in order to find these new therapies for our patients, we're going to have to test a lot of compounds. And our way of testing these compounds means that we can accelerate the development of the ones that are really going to be efficacious. And this just saves a lot of time for the development process, but also it means that our patients would have access to more therapies faster.